Okay, next we're going to discuss how to read the codon table and the codon wheel. We talked about the mRNAs, the mRNA triplets. Each 3 bases would code for a specific amino acid. So those are called codons. Each little sequence of 3 bases is a codon for a specific amino acid. So it says how to read the codon table. Using protein synthesis for translating the mRNA code into the amino acid sequence, and there's that translation again. That's the second step. It takes place at the ribosome. So you need a sequence of mRNA. Where does that come from? Remember, our mRNA came from transcription. The DNA is transcribed into messenger RNA, and that's because the DNA cannot leave the nucleus, but it has the information for making proteins. Well, the ribosomes are the ones that are going to be making proteins, and they're out in the cytoplasm, some of them attached to the rough ER. But the message has to get to them. The DNA cannot leave the nucleus. So it has to use a messenger to get those instructions out to those ribosomes, and that's the mRNA. So the mRNA is made in the nucleus, because that's where the DNA is. Little sections of the DNA are opened up, and the RNA polymerase comes in and matches those complementary bases with that section of the DNA. And remember, when we're talking about RNA, there are no T's in the RNA, so if there's an A and you think you're supposed to put a T, but it's RNA you're making, you're going to put a U. So, uracil. A is matched with uracil in the RNA. G's are still with C's. If it's a T, it'll be an A. If it's an A, there's going to be a U. But G's and C's are always together. And for example, here if we have our DNA sequence TAC, GGA, CAT, AAC, ACC, TGC, ATC, that would give us T would go to A would go to U, C would go to G, G to C, G to C, A to U, C to G, A to U, T to A to U, A to U, C to G, A to U, C to G, C to G, T to A, G to C, C to G, A to U, T to A, and finally C to G. The transcription, the mRNA sequence leaves the nucleus after transcription. After it's made from that DNA template, it's going to squeeze through those pores in that nuclear membrane. It's going to leave the nucleus and travel into the cytoplasm. It's going to find maybe a free-floating ribosome, or a ribosome that's attached to the rough ER. And it will attach to that ribosome and begin the second step of protein synthesis. It is called translation. At that point, it's going to have to use tRNA. tRNA is what I call tow truck, because it picks up those amino acids and it brings it back in to the ribosome. So it's like a tow truck. It has what's called an anticodon, which is complementary to the codons on the mRNA. So that tRNA that's got a specific amino acid attached to it, it's going to come in, and if it matches up complementary to that mRNA strand, then it's going to attach. And then it's going to join its amino acid to the amino acids there, and it's by peptide bonds, and it's going to make a long chain amino acid or a protein. That's what translation is. It's translating that DNA language into the amino acid protein language. So mRNA is read as a series of codons, again 3 letters for each codon, within the ribosome. tRNA molecules have an anticodon sequence of letters that are complements to the mRNA, and that allows them to attach to it. That allows the tRNA to come and bond with the mRNA to bring in those specific amino acids. Again, if the codon is CGA, then the anticodon would be GCU. If the codon is UCC, the anticodon on the tRNA would be AGG. U would go to A, C would go to G, C would go to G. This is kind of how it works down here. So amino acids, specific amino acids on that specific tRNA, and then it matches up to the codon on the mRNA. So now we get to the codon table. Okay. You're going to locate the first letter of your codon, and it's going to be the codon. This is a codon table, not an anti-codon table. It's the codon table. So you're going to use those 3 letters. That codon from the mRNA is always what you're going to take to the codon table or the codon wheel. It's going to be the mRNA sequence that you're looking for on that codon table or that codon wheel. So AUG, that's a specific one. AUG is the start codon. It's the one that tells the ribosome start making a protein. So AUG, you're going to find the first letter on the side here. This is the codon table. And we tend to see it more on tests at the codon table. So the first letter, you're going to find the first letter here. Well, AUG, that's the start codon. We remember that because school usually starts in August. So AUG, August, start codon, start making that protein. First letter is A, so we're going to be in this section here. The second letter is U. Well, that narrows it down to these right here. And then the third letter is G, so find the G and come straight across here. And that's going to be methionine. And methionine would be that start codon. That would be the amino acid that would start the protein. So AUG is the start codon. For all protein synthesis, AUG is going to be the start codon. So the first amino acid is going to be the methionine. A, U, G. So we just do that first letter, and then we go to our second letter. It narrows it down to the box again. And those are the ones in that box. And then the G tells us specifically which one in that box. We're going to come straight across in that G, and that's going to be the methionine. So let's try another codon. Okay, don't peek until you come up with the answer. Oops, and that didn't show up there. But histidine, so let's find histidine. And we can see if we can go backwards. And here's histidine. Maybe it was this histidine. If it's this histidine, then our first letter would have been a C. Our second letter would have been an A. And our third letter would have been an E. So what do these codons have to do with proteins? Each codon represents an amino acid that will eventually form a protein that is used within the cell. So proteins are made up of hundreds of amino acids in a specific sequence. And it is that specific sequence of those amino acids that give the protein its shape. And it's the shape of the protein that determines its function. So proteins are made up of hundreds of amino acids in a specific sequence to make it a specific protein to do a specific job. Sometimes something happens and maybe an amino acid, the proofreading doesn't work out or whatever, the amino acids get out of order, or maybe one's switched, maybe a base got copied wrong when the mRNA is getting made, or when the DNA replicated, maybe a base got copied wrong. So that would mess up a codon and might end up putting a wrong amino acid in there. But these are called mutations. When something happens and gets those amino acids out of order, or those bases are in the wrong order on the DNA or the mRNA, then that's called a mutation. So here's a long string. It might code for a glycine, and then a isoleucine, and then a valine, and then a cysteine, and then a glutamine. Okay. And the specific order will determine what that protein is going to do. So long strings of amino acids form proteins. In one particular case is a hemoglobin molecule. Notice there's some bonds up here. Those also determine how it's going to fold. When we talked about the organelles, we talked about ribosomes on the rough ER, how they'll make proteins. And then those proteins will go into that rough ER, and as they're going through that rough ER, they get folded into shape. And when that happens, that's part of the process, is it's going through the rough ER. And it gets pinched off and goes to the Golgi and gets modified even more. So they're folded depending on which amino acids they can make bonds within those proteins. It causes it to bend a certain way, and the way that it's bent determines its job or how it's going to work. Like hemoglobin in red blood cells, it has to be shaped just right to be able to carry an oxygen molecule and then pick up carbon dioxide molecules. So it's all determined by that sequence of amino acids.